Welcome back. Today we are going to continue our discussion on genetics, and we're going to look at something called Mendelian genetics. And Mendelian genetics is kind of the simplified version of genetic inheritance pathways that you'll evaluate in this course. And what we're going to do here is just briefly talk about Gregor Mendel and some of his preliminary experiments that allowed him to come up with some of these ideas: the law of independent assortment, law of segregation, that kind of dictates how traits, alleles are inherited through different generations. So again, we're going to talk about Mendelian genetics. Mendelian genetics is going to be contrasted with non-Mendelian genetics. And non-Mendelian genetics uses ideas and concepts that are more complex. So in Mendelian genetics, one allele is dominant and one allele is recessive. Right, and think back to our previous discussion on alleles, right, where we talked about alleles are just versions of a gene or versions of a trait that code for a particular protein. So we kind of discussed alleles and where you find them on chromosomes. And again, they're just modified versions of one another, and they can change how a trait appears, change its phenotype, right? And we use these terms heterozygous, homozygous, dominant, homozygous, recessive, terms of genotype and phenotype. We've defined all those things already up to this point. You're going to see those come back into play here. So what we'll do is we'll bring in Mendel, and here's Mendel, Gregor Mendel. He was a monk that lived in the 1800s near Austria, and he started growing pea plants in his garden. And he started to observe which traits get passed on at what frequency. So why pea plants? Well, pea plants, they have a lot of observable characteristics like flower color, height, seed shape or pea shape, round versus smooth, or round versus wrinkled, pod shape, constricted versus smooth, tall versus short, axial versus terminal flower position. So lots of different variations on this plant that were easily observed, a very short generation time, meaning he could grow them in a really short window. And they were just easy to work with. They grew well in Austria, in the area of Europe that he lived in. So that's what he was utilizing. And what he would do is he would take true breeding, so we're going to use this word, true breeding pea plants of one variety, and cross them or breed them with true breeding pea plants of another variety. So we're going to set that up, just kind of look at how that works. So let's say we take a cell from the purple plant. So this is a true breeding purple flowered plant. And you're going to see this word true breeding quite a bit. True breeding means that this plant is homozygous. Homozygous. And Mendel, he would not know if this purple allele was dominant over white, the other version of the allele. Now we know that because of his experiments, but back in the time he did not know that. So we're just going to use this symbol. We're going to say big A is purple and little a is white. Now Mendel, again, did not have the luxury of knowing which one is dominant, which is recessive. So we could use a P and a W for purple and white. But to keep it simple, we're just going to use a big A and a little a. And we know now that purple is dominant over white. Mendel did not have that luxury. Okay, so there are, I believe, 17 chromosomes in a pea plant. And we are going to look at, here's the nucleus, here's the DNA. What we are going to do is we are going to pull out this chromosome. We're going to pull out chromosome number 7. And we're going to look at where this allele for flower color resides. So flowers, they have male portions and they have female portions. and they can crossbreed themselves or breed themselves, or they can crossbreed other plants. So what we're going to do is we're going to look at the pollen grain that gave rise to this seed that grew this plant. And we're going to look at chromosome number 7. So this is the pollen, the male portion of the plant. So we're going to look at the male chromosome that this plant would have received from the pollen that formed it. And we're going to say that because it's purple, it's going to have the purple allele. So a big A. We're also going to look at the ovule, or the female portion of this plant that was pollinated by that pollen. And it's going to have, again, a set of chromosomes as well. And because it's a true breeder, this true breeder, we could assume that it's going to also have that dominant allele big A. And what Mendel would have had to have done is grown purple plants for a very long time, multiple generations, and seen that only purple flowers came about from those particular breeding events. So we call that a true breeding flower. So Mendel's got these purple plants that come from only other purple flowers. And he is going to cross, or he's going to breed them. So he's going to cross them or breed them. And we'll show you how he's going to do that. So let's bring in kind of our setup as far as how this works. So when you crossbreed plants, this is what you have to do. You've got to remove the male portion from one plant. So this over here on the right would be like our white plant, even though it's purple petaled. Can just imagine that it's white for a moment. And he's going to be removing the male portion, the anther where the pollen is produced. So he's got a white flower that has just the female portion, just the stigma, where the ovules are that are eventually going to give rise to small peas. And then that'll be the offspring. So he's going to take a white flower like that, and he's going then to take the pollen from a purple flower on the anther, these little brown things, paint them onto a brush, the pollen, and then he's going to brush that pollen into the female portion of the white plant, acting as a pollinator. And that is effectively then crossbreeding those plants. So he's preventing this plant from self-fertilizing because he's removed the anthers, and he's effectively then breeding that plant. And he will know that then he controlled that cross because it doesn't have the capacity to self-fertilize. So that's how he's setting this up and how he's doing this. It's a very elegant experiment for the time. So what we'll say is he will take, so he's got these true breeding purple flowers. He's then going to take these true breeding white flowers, and again, of course, they are homozygous, but they are homozygous with the white allele this time. So we're going to sketch out those chromosomes. So here is the male pollen portion from this white plant. So again, male number 7. And then we'll have the female where the ovule would be on this white plant that came from its parent. And again, it's a white plant, so it's got only the recessive, only the little a versions of the allele. So this is what we're crossing. We're crossing a purple flower, big A, with a white flower, little a. And that's how Mendel began his experiments. He did this for all these other traits as well: height, flower position, pod texture, seed texture, seed color, green versus yellow, all these sorts of different things that could arise. Okay. And what we do here is something that you're probably quite familiar with. It's called a monohybrid cross. And we're going to set this up and solve this for you so you know how to execute these problems. So a monohybrid cross, better known as a Punnett square, right? You've undoubtedly done these probably in 7th grade. And what we want to do first is we just want to define our alleles. So we're going to say that the big A is our purple allele and our little a is our white allele, right? Because again, Mendel didn't have that luxury, but we do. So we're going to map it out here. We want to then define our parental generation. So parental generation, or P generation. And a parental generation, if we look back, we had a purple flower and a white flower. So we're going to have this purple flower here. So this is parent number one. So purple. And we're going to cross that with a white. And again, they're homozygous dominant and homozygous recessive. So this is our parental generation. And what we want to do then is breed those, and we're going to come up with what's called an F1. An F1, F stands for filial. And this is where we start setting up Punnett squares. And you just draw the Punnett square. One parent goes to the side, so over here, and one parent goes up top, over here. Now, why do we do a Punnett square? Well, a Punnett square brings into play the concepts that we looked at in meiosis. It brings into play this independent assortment idea that these chromosomes, when gametes form pollen or ovules, they could arrange themselves, the 17 pairs of chromosomes could arrange themselves, or the pairs of chromosomes can arrange themselves in any different order here as far as male versus female contributions on the left or the right. So we have to consider multiple options, not just one box, but multiple boxes, depending on what traits we're looking at. And then we also have to take the alleles apart from one another, the law of segregation. So we're really modeling independent assortment, and we're modeling the law of segregation here. Because independent assortment, we've got to consider these various different arrangements. Again, we're just looking at one particular trait here. If we looked at more traits, we would need to look at more combinations. And we're also then pulling the alleles from one another because of that separation. So again, this models the law of segregation, Mendel's laws. And that's why we separate the alleles here like we are. So we simply bring in the alleles as they recombine in offspring. And this doesn't mean the plant has 4 offspring. It means these are the odds. This is the odd set that occurs for that offspring. So what do we have here when we start looking at this F1 generation? We have a 100%, all of them are purple and none of them are white. So we can create 2 different ratios. We create a genotypic ratio, which is a comparison of the genotypes or the symbols. So we have 1, 2, 3, and 4 big A, little a genotypes, and 0 anything else. So that's our genotypic ratio. And then we also have something called a phenotypic ratio. Phenotypic ratio. So phenotypic ratio is a ratio of what they look like. So purple, 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 because they all have the dominant allele. So we have 4 purple and 0 anything else. So this is what Mendel started to see over and over and over again with all of those various characteristics. A 4-to-0 ratio, they would all be that dominant version of the phenotype. And you can figure out which traits were then dominant. So the next stage, the next phase for Mendel would be to develop an F2. An F2 is done by taking members of your F1. So he's going to take individuals from this F1, which all have the same genotype. They all are purple, and he's going to breed them. So we've got big A, little a, crossed with big A, little a. Again, this is a purple and a purple. And again, setting these up in an organized fashion is the key to solving these problems properly and correctly with accuracy. So we set up our Punnett square. Go ahead and put the parents in their respected positions. So we have big A, little a, and big A, little a. We started then looking at our odds that are possible. And we see this classic ratio that comes from Mendelian genetics, a 1-to-2-to-1 genotypic ratio and a 3-to-1 phenotypic ratio. And we see this all the time when you have 2 heterozygous individuals bred for a particular trait. And Mendel saw this, again, with all of the different traits that he was testing for. You know, if he grew 1,000 plants from 2 of the F1s, he would see that 750 of them would exhibit a purple phenotype, and 250 or 25% of them would exhibit a white phenotype. And you saw that over and over and over again. So we're going to look at our genotypic ratio is a 1 big A, right? 1 big A, 2 big A, and then 1 little a. So a 1 to 2 to 1 genotypic ratio. And then we have our phenotypic ratio, which is a 1, 2, 3, a 3 purple, and a 1 white. So 1, 2, 3 purple, 1 white. So 75%, 3 out of 4 are purple, 25% are white. Now that doesn't mean that they have 4 offspring. It means that these are the odds of offspring. So there's a 75% chance of getting a purple flower and a 25% chance of getting a white flower. And again, this is the method that you want to use to solve these problems. So let's talk about one last thing that you can do with this simple setup before we look at more complex ideas in future videos. We're going to look at a test cross. A test cross. Test cross. So let's say that we've got a purple plant or a purple flower. Purple flowering plant. Right? And we don't know what the genotype is. It could be big A, or it could be big A, little a. It could be either of those. And we want to know for the purposes of doing a genetic test, a genetic experiment. So you can perform what's called a test cross, where you are going to cross an unknown genotype to a known genotype. We call that known genotype in this case is going to be that little a. It's going to be a homozygous recessive. And we're choosing that individual because we know what the genotype must be. It's got to have both recessive alleles if it's white and not purple. If it had a big A, it would be purple. So we know the white one has to be little a. So this is what this test cross looks like. Here's our parental generation. We have the unknown that we don't know here. Again, this one is purple. That's the one that we want to know what the identity is. And then here is our little a, our white one, our homozygous recessive that we're using for our test cross. So we've got two potential outcomes of this cross. So we're going to just kind of break our page in half here, and we're going to look at these outcomes. So outcome one is we have big A. So again, this is our F1. We have big A, and then we don't know what that is yet. Cross with little a. And then we have the same thing over here, big A. We don't know what that is yet. Cross with little a. So let's consider the possibility that this is big A, that the parent is homozygous dominant, a true breeder. So we can see where this goes, big A, little a, and we get all purple offspring. So if you were to do this test cross and you got 100% purple offspring, you grew 1,000 plants and they were all purple, you would know the identity of that individual up here would have to be homozygous dominant. On the other hand, if you did a test cross and consider this being heterozygous, you would get 50% that are purple, so half that are purple, and 50% here that are white. Okay, so if you did this test cross, you grew 1,000 plants, 500 are purple, 500 are white, you would know definitively that that individual up here would have been heterozygous and not homozygous dominant. So a test cross allows us to work backwards and kind of fill in genotypes that we're not sure of. So in the next video clip, we'll look at what happens when you're dealing with two alleles on separate chromosomes, and then we'll also look at what happens when we're dealing with two alleles that are on the same chromosome. So we start adding layers of complexity. Then we'll get into non-Mendelian inheritance patterns, where we'll look at things that really don't adhere to this dominance type of model. Okay, so I hope that's helpful, and I'll see you next time.